Hello, Arnab, can you hear me?
Yes, I can hear you.
We will wait for a few minutes. We will wait for our participants to join. Kindly bear with us.
Okay.
Thank you. Hello, Arnab. How are you?
Good. How are you?
I'm fine. Thanks for joining us.
Yeah, thank you.
I think we should get started by the time the participants will join.
Okay.
So, greetings to one and all. It gives me an immense warmth and great pleasure to grace all of your presence in the interests of the entire soil community. It gives me tremendous contentment to be moderating the current series of webinar funded by National Agricultural Higher Education Project of Coast Kashmir. Before we begin this seminar, I would like to express my heartfelt gratitude to all of you who sincerely committed to this World Soil Day event to make it a big success. This event is impossible without the support of each and everyone present here. These events are organized and conducted for the general audience to gain insight and information that would remain effective in their lives. Topic for today's webinar, soil microbial metabolism for sustainable and balanced nutrient management, was chosen keeping in mind the theme of this year's World Soil Day. Now, before we get started, just a little housekeeping as usual. If you have any questions during the presentation, please type them into the QA box only. We will have the time for QA session at the end. To keep the quality of sound functional during the meeting, please mute yourself. Now, may I request Dr. Mumtaz Ganai to please introduce our esteemed speaker to the audience.
Thank you, Dr. Shabir Ahmad Bangru Saab, for giving me an opportunity, or I can say that this is a pleasure for me to introduce young, dynamic, dedicated, and talented soil scientist who is basically from India, I think. I welcome him first, and who is named as Dr. Arnab Bhowmik, who is Assistant Professor Soil Science and Soil Microbiology in the Department of Natural Resource and Environmental Design, North Carolina A&T State University, USA. Dr. Bhowmik has completed BSc Agriculture from Bidhan Chandra Agriculture University, West Bengal in 2010. I think he's my batchmate because I have also completed my bachelor's in 2010. Now, he has qualified ICR GRF in soil science with All India rank 4 and completed master's in agriculture in soil science and Punjab Agriculture University, Ludhiana. Dr. Bhowmik pursued doctorate degree in soil science in 2016. Same for me here, from North Dakota State University, where he was inducted to Gamma Sigma Delta, which is the honor society of agriculture in US. Afterwards, he worked as a postdoctorate fellow at the Pennsylvania State University, collaborating on USDA and NIFA-funded projects to study aspects of biological soil health management for climate-adaptive agroecosystems. His current research lab at NCAT focuses on soil microbial and biogeochemical natural nutrient cycling, mainly carbon and nitrogen, biological soil health indicators, and greenhouse gas emission. Not only this, Dr. Bhowmik has won some awards, which include gold medal at master's level, distinguished graduate students award during PhD. During his first 2 years as a tenure track faculty, he was successful in securing about $2.3 million in research funding, for which he was named University Rookie Research of the Year. And this, I think, this is a big thing for a scientist, for any scientist in his early career. He currently serves as the chair for student mentoring program for soil and water management and conservation division, and as a member of the Clerk Soil Biology Graduate Student Scholarship Committee. His research has been presented in Soil Science Society of America, America Geophysical Union, Ecological Society of America, and American Society for Microbiology meetings. And this is a big thing he has achieved. Now, I would like to handle control to Dr. Arnab Bhowmik for his presentation on soil microbial metabolism for sustainable and balanced nutrient management. Dr. Arnab, please.
Thank you very much. Let me try and share my screen. Can you see the screen?
Just being uploaded.
Yes.
Okay. And is my voice audible?
Yes, perfect.
Okay, thank you very much. So, good morning, good afternoon, good evening, depending on where you are in the globe. For me, it's around 8 a.m. here. And I would like to start off by saying thank you very much for this opportunity, and I'm glad to be here. So, like you mentioned, today's topic would be soil microbial metabolism for sustainable and balanced nutrient management. So, just to make things clear, and just to make sure that we address the wide range of audience we have today, I would like to give a brief introduction on soil health. Most of you know about soil health. So, soil health is an integral part of sustainable agriculture. Soil health is a holistic approach, a whole unifying approach, which is defined as the sustained capacity of living soils to function. Now, see, this is different than what we usually used to look into soils. We always look into soils as a medium for plant growth, which is super important. But at the same time, soil health unified the concepts of not only the plant health but also animal, human, and environmental health. So you have all those components of the ecosystem brought together in this holistic umbrella of soil health. Now, soil health measurement started a long time back. It started with measuring traditional soil fertility. So this would be, when you give your samples, do chemical testing on your samples to determine the amount of nutrients present in the soil. With time, however, we realized that the chemical components of soil are important, but also we need to keep in mind the biological components, the dynamic components of the soil. So, for example, when you supply any kind of nutrient to the soil, it interacts with the living soil and it undergoes a lot of, let's say, biogeochemical changes. And what happens as a result of that change is a part of the nutrients is assimilated by the biota. Now, by the biota, I mean, plants or microorganisms or any kind of organisms. The part which is not assimilated could be lost into the environment, and that could have severe environmental impacts. So, an example of that would be, nitrate leaching and causing eutrophication, or let's say, greenhouse gas emissions into the atmosphere, raising the temperatures. So just to make sure, where my research fits in this whole picture of soil health is in managing the biological soil health components, and this is in order to better understand the biological nutrient cycling, mainly carbon and nitrogen, which is related to soil fertility and the environmental impacts. So we need to make sure that we understand the importance of microorganisms in the soil, because when we talk about soil health, we are talking about the health of the microorganisms along with the soil. So microbes are the key players. Now, any kind of soil health management practices are directed towards in improving or increasing the amount of soil organic carbon or soil organic matter in the soil. So management practices like incorporation of cover crops, crop rotations, animal amendments, or even reduced tillage, they increase organic matter in the soil. Now, in organic matter, the carbon and nitrogen is coupled, which means that it comes together. What happens is microorganisms, which are heterotrophic, they decompose this organic matter. And by the process of mineralization, they release ammonium. Now, during this process, carbon dioxide is emitted as a product. Now, again, we all know carbon dioxide is a greenhouse gas. Now, what happens to ammonium once it's formed by the mineralization process? Ammonium under aerobic condition, which means that when there is enough oxygen in the soil, could be converted into nitrate. Now, this is a microbial process. This is called nitrification. And the rate limiting step in this process is regulated by AMOA gene, which in turn regulates the ammonia monooxygenase enzyme. So it's a highly microbial derived or mediated process. Now, one of the intermediate products or byproducts, I should say, in the nitrification process is nitrous oxide. Now, we all know nitrous oxide is a greenhouse gas, and more importantly, it's 300 times more potent as carbon dioxide. So once nitrate is formed, what are the fates of nitrate, possible fates? One of the possible fate could be, nitrate leaching from the soil, or, take up by the plant uptake, right? The other fates could be immobilization, which is a process by which nitrate is assimilated by the microorganisms and becomes a part of the microbial cell. So that is a process of immobilization. Or if there is not enough oxygen in the soil, so nitrate could be denitrified to nitrogen gas, inert nitrogen gas, the gas which is in the atmosphere, therefore closing the cycle. Now, denitrification, I would like to say, is a very tricky process. It's tricky because, if you don't have the right amount of carbon, nitrogen, and oxygen in the soil, the end product of denitrification could be nitrous oxide instead of nitrogen gas. Now, again, like I mentioned, nitrous oxide is a more potent greenhouse gas. And again, this last step where nitrous oxide is getting converted to nitrogen, this critical step is regulated by microorganisms with the nitrous oxide reductase enzyme, which is regulated again by this gene that I mentioned here, NOS-Z. So these are the important parameters and important controls which kind of regulates not only the nitrogen cycling, but also the coupling of the carbon and the nitrogen cycling. So this is the map of United States. So what I'm going to do today is kind of briefly discuss about the range of experiments and range of management practices that I have worked on in the United States, which not only represents different climates, but also different agroecological regions, along with different management treatments. So the first site, and all of these are long-term experimental sites, and we all know long-term experimental sites are expensive because you need continuous funding to keep on this experiment. So the first site was located in North Dakota, and this was affiliated with North Dakota State University. And here we were looking into two different contrasting treatments, conventional till and no-till. So conventional till would be something like moldboard plowing, and then no-till is reduced, no disturbance in the soil. And these experiments were 3 years old. So it has been only 3 years since the difference in the tillage intensities were implemented in these experiments. The second set of long-term experiments were located in, Washington, and these were affiliated with Washington State University. And again, this was a 12-year-long experiment. So it was 12 years since they made the amendments, which are the treatments here. So they had 3 different treatments: compost, broiler litter, pasture, along with reduced till or no till. Now the important thing to note over here is the difference in the quality of organic matter which was added for those 12 long years. So we were very interested to see how long-term management actually impacts soil health. The third set of experiments were located in Pennsylvania. These were affiliated with the Pennsylvania State University, or Penn State. And here we were looking into the effect of different types of manure and manure application methods on soil health. So we had broadcast manure, or we had synthetic fertilizer, and we had broadcast manure with tillage. So you'll see there is a wide range of management practices in these 3 different long-term experiments, along with different climate conditions, because climate plays a huge role in determining, the soil microorganisms and their metabolisms in the soil. I would also like to mention that, we conducted experiments at all 3 different scales. The field scale, where we went into the field and took measurements, gas measurements, or, soil measurements. We also did lab incubation experiments where we collected the soils from, those long-term experiments, brought it back to the lab, and then manipulated the conditions and conducted the incubation experiments. We also conducted experiments at the molecular scale, which means that we extracted DNA, and then we analyzed the microbial communities, or the abundance. And I'm going to discuss about that briefly today. So what did we measure, right? what we measured was the microbiologically regulated soil carbon and nitrogen cycling processes, which are also potential biological indicators of soil health. So basically, the nitrogen cycle that I explained to you, most of the components of those cycle we measured. So for example, we measured the active carbon and nitrogen fractions you see over here. We measured the carbon dioxide, we measured ammonium, therefore we measured mineralization rates. We also measured nitrate, therefore we measured nitrification rates, and also denitrification when we measured nitrous oxide. So along with the processes, we also measured or identified and quantified the microorganisms which are involved in these steps. So specifically, we were interested in the MOA gene and the NOS-Z gene, which are functional genes. And in order to quantify these genes, we used a quantitative PCR or a real-time PCR. Now, I'm going to discuss, because of time, I'm going to discuss about a couple of specific experiments, which I think is important, to share because of, what we learned from the results. So this particular experiment was an incubation experiment where we wanted to measure, the components of the nitrogen cycling that I just mentioned in my previous slide. So this was a soil microcosm setup, experimental setup, where we were interested to measure greenhouse gases. And at the same time, we wanted to see the change in the microorganisms, the quantity of microorganisms, specific microorganisms in the soil during the incubation. So what we did for this purpose is, we had so many different types of soils, right? Not types of soils, but wide range of management from different parts of the country. So we collected soils from those wide range of management. I call it long-term management history. We brought it to the lab, and then we manipulated the moisture and the amendments. Now I call it short-term management. Now for moisture, we maintained 40, 60, and 80% water fill pore space. So for if you think it in terms of how it relates to the field conditions, water fill pore space, which actually controls the amount of water, is a proxy for the amount of oxygen present in the soil, right? So therefore, what happens is, by regulating the water fill pore space, you are actually modifying the oxygen and therefore the microbial metabolisms involved in the nitrogen cycle. And 40, 60, and 80, why these specific water fill pore space? Because we wanted to look at the range, okay, because of let's say different intensity of rainfall. Okay, so 80% would be a heavy rainfall, 40% would be a light rainfall. So, and also 40, 60, and 80 are very critical for the nitrogen cycling because it denotes a shift from one process to the other. Now, for the amendments, we added two different types of amendments. We either added urea or we added beet tops. Now, urea, like everyone knows, it's a chemical fertilizer. Therefore, we wanted to see what happens if we add chemical fertilizer to the soil. And beet tops was to simulate the effect of leguminous cover crop incorporation in the soil. And again, these are two different strategies of fertilization, inorganic versus organic. And I would like to mention that both of these were N15 labeled because we wanted to track the nitrogen in the nitrogen cycle. And the temperature which we conducted this experiment, you can see this incubator, it was a huge experiment and huge number of samples. The temperature was, we wanted to simulate late fall to early spring. So, in Indian conditions, it would be something like the winter conditions when we have the cash crop already harvested. And then, we have a lag before the next spring or let's say next summer crop. So the reason why we chose this window is because, the general notion among the growers is that since the temperature is low, not much microbial activity is going in the soil. So you can leave the soil fallow, or you can do anything you want. You can have cover crops, or but we wanted to focus on that in order to see, what actually is happening in that period when you don't have a cash crop, usually don't have a cash crop. Okay, so what did we find? This particular diagram is a non-metric multidimensional scaling. So it's a non-parametric method of analyzing data. In simple words, the points which are close to each other are similar. The points which are far away from each other are different. Okay, and I'll try to simplify it because there's too much of stuff going on in here. So what we found out is from the incubation experiment, the soils where urea was added, we found an accumulation of ammonium in those soils. Okay, which tells us two things. First thing, the urea we applied had amide nitrogen, right? So it converted into ammonium. That's fine. The second thing, which is more important, is that ammonium did not get converted into nitrate. That is why we had an accumulation of ammonium if you look into the nitrogen cycle, right? Now, why did that happen? That happened because nitrifiers, which are supposed to convert ammonium to nitrate, are very sensitive to low temperatures. Okay, so it's true to some extent that if the temperature is low, nitrifiers are not very active in the soil. However, when we looked into the treatments where we added beet top residues, again, beet top residues was to simulate the effect of leguminous cover crop incorporation in the soil. We found a huge amount of nitrous oxide and carbon dioxide emitted from the soils. Okay, so the first question was, in order to have nitrous oxide, if you look at this nitrogen cycle over here on the right, in order to have nitrous oxide, you need nitrate, right? But if there is no nitrification, where is this nitrate coming from? And therefore, where is this nitrous oxide coming from? That is very strange. So what we found out, and the N15 labeling actually helped in this case because we were able to track that nitrogen. What we found out was the beet top residues or the leguminous cover crop actually had nitrate present in the tissues. Okay, so what happened is when we incorporated the cover crops into the soil, that nitrate in the tissue kind of came out into the soil. And even at low temperatures, denitrification occurred. So we found out that denitrifiers are not as sensitive to the low temperatures. So the take home message from this result is, during that low temperature conditions, when usually you would not expect to have a crop, if there is nitrate present in the soil, that would get lost. It could get lost depending on the soil texture. It could get lost by leaching, or it could get lost by the process of denitrification as nitrous oxide. And again, nitrous oxide is a greenhouse gas. So now the question is, okay, how can we prevent that loss of nitrate, right? That's a solution. We are looking for a solution. So if you remember, I mentioned for North Dakota soils, we had 3 years of that tillage effect, right? Conventional till versus no-till. In this particular figure, in the y-axis, you have global warming potential, which kind of takes into account both carbon dioxide and nitrous oxide. So what we found out, strangely, is even after 3 years of no-till, we could not see any significant effect in the global warming potential. Or in other words, the amount of carbon dioxide and nitrous oxide was statistically similar for the both tillage treatments. However, when we looked into the other set of experiments, which was in Washington, if you remember the 12 years long tillage and amendment effect at Washington, we found out that the soils which were incorporated with compost and reduced tillage, there was significantly lower amount of global warming potential, which was really exciting because that is what we want. We want the greenhouse gas emissions from these amended soils to be lower. So now the question is why? So in order to answer that why, we started looking into the amount of carbon that was present in the soil. So in the left-hand part, if you look over here, you'll see that there was no significant difference between the amount of carbon between the conventional till and the no-till. which tells us that 3 years is not sufficient to significantly increase the amount of carbon in the soil so that you can have added benefits in terms of reduced greenhouse gas emission. However, when you look in the Washington sites, which were again, I'm emphasizing on the time, 12 years, that's a long time. So that 12 years of difference in tillage and amendment actually increased significantly the amount of carbon in the composted plots. Okay. Now, what that tells us is that extra carbon actually helped to retain the nitrate, okay, by the process of immobilization, and therefore reduced the global warming potential. So in brief, how can we reduce nitrate loss during that time is by increasing or building that carbon pool or carbon bank in the soil. Okay, so we're also interested in looking into different fractions of carbon. There are a lot of literature, in different types of fractions: physical, chemical, biological. We decided to stick with a three-pool non-linear model. Okay, and this model actually gives you defines three different types of pool. The first pool is active pool. So active pool would be something, let's say, the carbon which turns over in a growing season, or in other words, this is the carbon which is very active or available to the microorganisms. The second pool is the slow pool. The slow pool of carbon is the carbon which is a bit more protected, and it takes maybe, let's say, months, several months to maybe some years to turn over. The third pool is the resistant pool, which is a very recalcitrant or stable pool. And this pool, takes thousands and thousands of years to turn over. And so what we found out was, for all these 5 different treatments, the amount of the percent of total carbon was 1%, which is really low as a percent of the total carbon. So which tells us that in these treatments, there was a lot of lower amount of active carbon in the soil. When we looked into the slow carbon pool, and this is the lab MRT, which means that mean residence time. So which is an estimate of the time it takes for that carbon pool to cycle. So this is the most interesting result we got from this table is if you look at the rates between the no tillage and the conventional tillage, you'll find that the no till has lower MRT. The no tillage has lower MRT, which means that the carbon in the no till was cycling faster than the carbon in the conventional till. Whereas if you look into the compost, the broiler litter, and the pasture amended plots, we found out that the pasture plots had slower. Pasture would be, grazing or, grassland. The carbon in the pasture amended or the pasture soils cycled slower than the compost or the broiler litter amended soils. Okay, now we always talk about total carbon, right? We need to increase total carbon, we need to increase organic carbon, but sometimes the total soil organic carbon is not a very good indicator which is sensitive to change in management, because you want to see that change. So suppose if you are transitioning from intensive tillage to reduced tillage, you want to see how that is making a change in the carbon, because carbon is very important. So what we decided to look into another pool, which is also known as the permanganate oxidizable carbon pool. And this is sometimes known as active carbon. It's sometimes known as reactive carbon. But basically what it means is it is that fraction of carbon, of total carbon, which the microbes can see and can have access to. So what we found out was this particular fraction was highly correlated with the carbon and nitrogen mineralization, which told us that this could be a very good indicator of the amount of carbon which the microorganisms are looking at. So And therefore, in order to have indicators of soil health, we can use this particular fraction in order to see the difference in the management. Okay, we also measured and some DNA based novel soil health indicators. So if you remember, I mentioned about the AMOA gene, right? So I'll briefly talk about what this is. So nitrification usually is thought to be a two-step process. The first step is the rate limiting step, and that step is regulated by ammonia monooxygenase enzyme, which is in turn regulated by the MOA gene. Now, there are two types of keystone species which can conduct this process: ammonia oxidizing archaea and ammonia oxidizing bacteria. Now, the reason I say keystone is because only these organisms can conduct this process, which means that if these organisms are not present in the soil, nitrification or uh, nitrification will not occur. Okay, so these are keystone species. So we strategically looked into these particular species. What we found out was the abundance or population of ammonia oxidizing archaea and ammonia oxidizing bacteria was sensitive to this change in the management, which is a huge thing, because when you think in terms of these tiny microorganisms, it's a huge deal according to me to I think to see differences when you're looking at a field scale, because sometimes microbial metabolisms are very tricky. So this was really exciting for on for to realize that we can even use DNA based novel soil health indicators to reflect the change in the management. So from this is the first part of my talk. So several take homes from here. The first take home is late fall or early spring, low soil temperature. Again, this would be the winter time in India. Limit nitrification, which is formation of nitrate, but nitrate in the cover crop residues can contribute significantly to nitrous oxide emissions through denitrification. So I just want to make clear, I'm not saying, you shouldn't have cover crops. What I'm saying is you need to think beyond that, which means that, it's good to have cover crops, but you also need to think about the type of cover crops you have. Do you have a lot of legumes? Because if you have a lot of legumes, there could be nitrate leaching or nitrate loss. Or do you have a good balance of, let's say grasses and legumes? So the cover crop mix is very important to consider, rather than just, having cover crops and, thinking, well, I have cover crops, my soil is protected. Well, you need to think deeper. The second take home is only reducing tillage without sufficient carbon addition for a long term do not build soil health or reduce greenhouse gas emissions. So we saw that 3 years of change in tillage didn't really reflect in building soil health, right? So the take home from here would be, with reducing tillage, we need to also provide sufficient amount of carbon. Now that could be, you could add any kind of organic amendment, or even you could increase the crop yield, right? Because if you increase the crop yield and then you return the residues back in the soil, that's building the carbon. So some of the things that you need to take in mind. And climate plays a very big role here, because climate regulates the yield in most of the places, the rainfall, the temperature. And third is, novel biological indicators like nitrogen cycling genes responded to management effects. Okay, so the second part of my talk, I'm going to talk about another soil health amendment, which is manure. And I'm going to talk about a specific process in the nitrogen cycle, which is called DNRA, which stands for dissimilatory nitrate reduction to ammonia. Now, why is this process even important? This process is important because if you look in this diagram, DNRA is conversion of nitrate back into ammonium. Okay. Now, how does this help? This helps because nitrate is mobile. It moves very fast in the soil. If you can convert it back into ammonium, you are actually making the nitrogen immobile, which is less prone to loss. The second thing is you are also reducing the tendency of this nitrate to get denitrified as nitrous oxide. Okay, so therefore facilitating this process could conserve nitrogen in soil-based ecosystems. Now, how do we measure this process? We can track this process by tracking the microorganisms. We can use it as a genetic marker. So like I have mentioned here, these organisms are called nitrite ammonifiers. From here on, I'm going to denote them as Na, nitrate ammonifiers. And they have this specific functional gene, NRFA, which can be used as a genetic marker, like I was mentioning, to track this process. Now, the interesting thing, what we found is manure is a source of this kind of bacteria. This is very cool because, what this means that if you're adding manure to the soil, you're also adding these groups of microorganisms, which has the potential to reduce nitrate back to ammonia, right? So very interesting. And we wanted to look into how different manure handling and storage techniques regulate these groups. So here in the US, I'm not sure about the India, but here in the US, a lot of farmers have anaerobic digesters where they convert the raw manure into digested manure. Now, it's called anaerobic digestion of manures. Now, the reason they do that is because it reduces the foul odor, which is associated with the raw manure. It also generates biogas, so they can use it to generate electricity. And the most two important things, it stabilizes the manure carbon and also increases the ammonium N content in the manure. So therefore, you can say that the digested manure would be more rich in plant available nitrogen as compared to the raw manure. So it's this conversion is helpful, but it takes some amount of money, a good amount of money, I should say, to establish the digester. So that's a constraint. But however, we wanted to see the effect of this anaerobic digestion process on nitrate ammonifying microbes. So for this, we collected 5 different, we sampled before and after manure digestion from 5 different farms in Pennsylvania. And we brought the samples back to the lab, and we measured physico-chemical properties. We also measured the microbial communities, and we did that by amplicon sequencing using Illumina MySeq platform. Okay, and very clear, distinct, and very interesting results. What we found out was the physico-chemical properties of the manure was impacted because of the digestion process. So if you look into this non-metric multidimensional scaling diagram, you'll see that the raw manure is a separate circle than the digested manure, which means that the physico-chemical properties were different after the digestion process. Not only the physico-chemical properties, but also the type of microbial communities in these 2 types of manure was different. So raw manure was different than digested manure. And if you look into these vectors, this arrow over here, you'll see that the carbon to nitrogen ratio is inclined towards the raw manure, which means that in raw manure, there is higher carbon to nitrogen ratio. And you all know, higher carbon to nitrogen ratio means slow decomposition. Whereas for digested manure, you'll see that there is higher amount of ammonium N, which is directed towards this vector, which meant, like I mentioned before, that digested manure contains higher, excuse me, higher amount of ammonium. And again, this is a heat map to kind of distinguish between the fact that the anaerobic digestion process impacts the type of microbial communities present in the raw versus the digested manure. So the next step was, okay, we see differences in raw and digested manure, right? What happens if we add that to the soil? Because soil itself has a different set of microbiome. So we are actually introducing 2 different microbiomes with each other, right? Raw and soil, digested and soil. So what we found out there is we did again Illumina sequencing, and we found out that the communities in the raw manure amended soils were different than the communities in the digested soil. Now, this was very interesting because it tells us that the difference which was present in the manure was transferred to the soil. So when we apply this manure to the soil, we can expect to see changes in the microbial communities. And we also measured the amount of nitrous oxide coming out from the soils. And we saw a significant difference there. We saw that the soil which was amended with digested manure had higher amount of nitrous oxide as compared to raw. Okay, so the next set of experiments, and I think this is close to the end. The next set of amendments I'm going to talk about is both cover crop and manure incorporation. So, we spoke about why cover crop is important, right? We spoke about how manure has the potential to regulate the nitrogen cycling. Now we'll talk about manure and cover crop addition. Now, this particular experiment was in Pennsylvania. It was a reduced organic systems experiment. So it was a certified organic experiment where we used two different sources of nitrogen for corn. The first one was a cover crop mix. So we had leguminous cover crop, which was hairy vetch, and then we had triticale. For the nitrogen from the manure part, we had raw dairy manure. So what happened is during the first 2 years, we found higher amounts of nitrous oxide emitted from the soils. And this was in collaboration with a modeling group, agroecosystem modeling group in at Penn State. And we were finding higher amounts of nitrous oxide from the soil, and we wanted to see what are the means to mitigate that nitrous oxide. Because see, we all know, we need to increase organic matter, right? So we need to add organic matter or organic manure or organic sources to the soil. But what we were finding is that addition of organic manure or cover crop is increasing nitrous oxide. So there should be a way to control that. So we asked two different questions. The first question was, do we know what is contributing to nitrous oxide? Is it the manure component or the cover crop component, which is actually contributing to higher nitrous oxide? The second question we asked was, do we know the mechanism? If you remember, I mentioned in the first part of my talk that nitrous oxide can be formed from both nitrification and denitrification process. So denitrification being the dominant source, but still we wanted to know the mechanism, what in this case is contributing. So for this, what we did was we manipulated a field, we designed a field microplot experiment to manipulate the addition and removal of cover crops and manure. So I'll show you an image. So in this first image, you see cover crop exclusion. So which means that we grew the cover crops and then we harvested it and we removed it. We did not incorporate that to the soil. So that was a cover crop exclusion. And then for manure exclusion, what we did was we did not incorporate or add manure to the soil. So we came up with this 4 different treatments: plus M plus CC, plus M minus CC, minus M plus CC, and minus M minus CC, which represents the 4 different combinations of adding cover crop or adding manure or not adding manure. Okay, so what did we find? We found out that avoiding the above ground cover crop incorporation caused 60% nitrous oxide reduction. Okay, so I'll explain it. If you look at this graph over here, you'll see that in the x-axis you have different combinations, the 4 combinations of manure and cover crop. And then in the y-axis you have cumulative nitrous oxide. If you look into this particular figure, in this particular treatment, you'll see that the amount of nitrous oxide in the plots where cover crop was not incorporated but manure was added had lower amounts of nitrous oxide versus the plots where both manure and cover crop was added. So it tells us that, removing that cover crop incorporation actually prevents a lot of nitrous oxide. So that's why, I say avoiding the above ground cover crop incorporation costs 60% nitrous oxide reduction with a 10% grain yield. Now you have to also equate the grain yield, or the yield, because cover crops are a source of carbon, nitrogen, and other nutrients too. So again, this emphasizes the importance of a cover crop mix, to know, how much proportion of legumes do you have in the soil, how much proportions of grasses do you have in the soil, because there could be a lot of times when having that right mix is going to really determine if you can get benefits or make that soil healthy. And for the mechanism part, we looked into isotopomer analysis. So this is um, looking into the intermolecular distribution of 15N in nitrous oxide. So again, I'm happy to go into the details for this process, but for the sake of time, we do a site preference. We do it using a mass spec, and based on that site preference values, we can determine what is the source of nitrous oxide. Is it nitrification or is it denitrification? And in this case, we found out that denitrification was the major source of nitrous oxide. Okay, so the take home from this part was manure digestion has certain benefits. Okay, so you can reduce the odor, you can generate electricity, but might contribute to increased nitrous oxide emissions. So there is a trade-off we need to consider. Manure digestion results in distinct shifts in the microbial communities and physico-chemical properties that has potential to regulate nitrogen losses from the soil. And then finally, leguminous cover crop incorporation into the soil might increase nitrous oxide emission. So very important point to keep in mind is what type of cover crop cocktail do you have, or cover crop mix do you have? Okay, so the overall take home message, ecology matters. So if you remember, I mentioned this diagram right at the starting of my talk. So therefore, what we know, or we have come to know from these results, is that the way you manage the soils is going to really matter how much nutrient can be assimilated by the biota, or how much nutrients can be potentially lost. Our aim as soil scientists is to manage the soils in a way that we can increase the assimilation by the plants, and at the same time reduce the environmental impacts. So the importance of a holistic approach or ecological approach is very important. So this specific diagram kind of points out to that. So for example, for a specific soil function to occur, you need three different types of things at the same time. So you need the right type of microorganisms, because if the microorganisms, they're the drivers, if they're not present, the process will not happen, whatever you do. But at the same time, you need the right soil management and climate, because just the presence of microorganisms is not going to make them work for that particular soil function. You need to have the right conditions, the right substrates, and again, those two things are going to be regulated by how you manage the soils and the climate in that area. So we so my program here at ANT, we have several focus areas. One of the focus area is industrial hemp, which we are looking into soil health management for CBD. Hemp CBD is a very I should say booming product in the US, and we are interested to see how we can increase the amount of CBD in hemp, but at the same time make it sustainable. I also have some projects related to nitrous oxide consumption process, so climate change, and how we can manage our soils to reduce the effect of greenhouse gas emissions, or looking into different combinations of cover crop and manure, how we can manipulate those in different climate and soil conditions, so that we can get the best out of that. And I would like to thank my funding agencies, the collaborating institutions, This is a snapshot of my lab website, so please feel free to visit. And I'm really glad to have this opportunity, and I think I we would be able to build a collaboration between the two universities at any capacity. So I'm really excited. So again, happy World Soil Day, and I would be happy to take questions. I don't know the format, but thank you very much.
Thank you, Arnab. Thank you. Really, it was an amazing presentation and excellent work. Let me share with the audience and with you, Arnab, that today ICR released it is State Agriculture University ranking, and our university ranks at number 9th among the state agriculture universities.
That's
So
awesome.
we would, yeah, thank you. So we would really love to have collaborations with you and with your university in future. And please share all your slides once we are done here. So before we proceed, I will request my colleague, Dr. Iqbal, if there are any questions. So he may select some questions and put Dr. Arnab, Dr. Iqbal.
Well, good morning to Arnab. Good evening to all of my colleagues who are listening to this beautiful presentation. There are a lot many queries regarding the subject we have talking in detail. One query is from Naik Ziauddin Rahman. He is a PhD scholar from Scotch Kashmir. He asks us, during a long-term management experiment, was there any biotic community that remained consistent or reflected least change in soil despite various treatments?
Yeah.
What?
So yeah, that's a very interesting question. The scope of this work was looking into only specific communities related to the nitrogen cycle, but we wanted to target, let me say, we wanted to target those specific microorganisms which we expect to change, because the whole point is, we are making these long-term changes in order to change the soil functions. And if we wanted to see if we can change that in those specific groups or specific drivers, microbial drivers. But there could be some communities which might be not impacted by the change in the management, and that would be especially generalists in the in the soil, which means that there are certain microorganisms which need to be present in, specifically for carbon cycling. They are always present there in irrespective of the management. But these specific microbes that I was talking about are the microbes which are very sensitive to those management changes.
Thank you. Another question is from Dr. Shahida Nisar. He asks us, uh, what could be the possible effect of urea application on residue decomposition, as it has been seen that spraying urea instead speeding up decomposition reduces the same. Has low temperature any role, sir?
Yeah, that's a good question. So we can think it about this in two different ways. One way is adding nitrogen, available nitrogen would actually increase the decomposition of the already stable carbon in the soil. So which means that we are reducing the amount of carbon sequestration in the soil. But another way to look at it is, if you're using or if you're adding urea, you're also at the same time increasing the crop yield. So if you can find a way to return that residue from the crop yield back to the soil, it kind of balances it off. But the thing with chemical fertilization is it's too quick. It's not really synchronized with the plant uptake. So we have a lot of available nitrogen sitting in the soil, which has like I showed, prone to get lost into the water or into the air by leaching or denitrification. So we have to be careful while doing that.
Another person, Ritik Chawla, is from lovely professional university, Punjab. He asks us, regarding the problem, we really listen this problem through our media channels, that pollution has increased in NCR. But and Delhi, there is some noise somewhere, please.
Okay, Eagle,
Kindly,
carry on.
kindly see, there is noise somewhere. Noise
Eagle,
is coming
carry
from
on.
somewhere, huh? There is a pollution problem in New Delhi and adjoining areas regarding the crop residue burning. He asked us, is there something scientific that scientific solution regarding the problem, so that we can manage it like decomposition and other things, so that
Yeah, I mean,
these crop produce may not be burned.
yeah, I mean, I can talk about the soil aspect of it. I'm not an atmospheric scientist, but I think one of the main reasons why the crop is burnt is because the growers are not aware what they can do with the residues. I think as as extension agents, or as outreach activities, we need to educate the farmers, and that is same for every parts in the world, not only in India. We as soil scientists, I think we need to tell them the benefits of storing that carbon back into the soil. And so I think soil health has been able to do that a lot, at least in the United States, because when you talk to a farmer and you say, well, you are degrading the health of the soil, so just think about your health. If you don't eat the balanced right nutrition, your health will be deteriorated, right? Same for soils. If you don't provide the food to the soil microorganisms, the health of the soil is going to deteriorate. And if the health deteriorates, it's not going to give you as much benefits, or it's not going to work as it used to in its right health. So the only way to improve the health is to provide the right kind of food, or to provide the right type of carbon amendments to the soil. So I think if we can translate that emotion to the farmers, I think a lot of these burning and would be decreased. But I'm a naive person to talk this, because I don't really deal with farmers, but that would be my approach.
Thank you for your nice presentation, Arnab. Back to Dr. Shabir.
Dr. Arnab, I thank you once again for such a nice presentation. Really, it was, I think our students might have taken a lot from your presentations. I thank you once again. Now, before we move out, I would request everybody to please open up their camera so that we can have a group photograph. I request everybody to please open up their camera. Okay, thank you guys. Thank you for joining us today. And thank you, Arnab, for such a nice presentation. Have a nice day. Thank you.
Yeah, thank you very much.
Thank you, Arnab.